Okay, this is your exam 3 study guide short answer section. So I'm just going to go through the first 2 questions actually in this video. Number 1 says, considered a diploid germ cell in an animal that is about to undergo meiosis to produce gametes. The diploid number of the cell is 20. How many pairs of homologous chromosomes does this cell have? Now keep in mind, a homologous chromosome means that you have chromosome number 1 from mom and you have chromosome number 1 from dad. Number 1 from dad, okay? So if you are a diploid number of 20, that means you have 10 pairs of chromosomes. So 10 sets, 1 from mom and 1 from dad. So how many pairs of the first one says, what is the haploid number for the cell? So the haploid number, if you would get rid of the pairs part of this question, the haploid number then would be 10. How many pairs of homologous chromosomes does it have? Again, that answer is 10 because 10 pairs. How many chromosomes would be present in each daughter cell at the end of metaphase 2? So this is a complicated thing, so let's talk about it a little bit. You've got this issue of metaphase 2 got rid of the tetrad, so you've got just these chromosomes going across. So each one of these is a sister chromatid, meaning you have a chromosome 1 here and 1's copy here. 2 here, 2's copy, 3 here, and 3's copy. All right, you follow me? It's important because I'm not going to give you the same numbers on your test, so you've got to really know this. So this is metaphase 2. Metaphase 2 now only has 10 sister chromatids total, but it's got 20 chromosomes total. Did that make sense? Because chromosome 1 and its copy, that's 2 chromosomes altogether. So you have 40 chromosomes total when you start this process, and then it's going to drop down and drop down again to where you get to 10. So 20 is the diploid number. When 20 doubles, you have 40. After the 40 splits, you have 20, and then it splits again, which brings you down to 10. So there you go. Next one. How many chromosomes will be present in each cell, or in each nuclei cell at telophase 1? So that's going to be the same as in each one at metaphase 2. It's still going to be the same as telophase 1 because you haven't split again yet. So once it splits again, then you end up with 6. How many tetrads will be present in metaphase 1? You will have chromosome number 1, chromosome number 1, chromosome number 2, chromosome number 2, and so on. So that's 10 tetrads because your 10 pairs are going to line up in tetrads. And the last one, will the daughter cells produced after meiosis 1 be diploid or haploid? At this point, they are haploid, excuse my bad writing, because you got rid of the homologous chromosomes at that point, and all you have left are sister chromatids, which means chromosome number 1 and its pair. So I know those numbers are a little confusing. If you need to rewatch that part a couple of times or look up a worksheet or something to help you get it, because like I said, you're not going to have the number 20 to start on your test. Next question, number 2. In the tongue-sole fish, I'm not even going to try to do its scientific name, sex is determined by a combination of genetics and environment. Genetically, male fish have 2 Z chromosomes, which makes them ZZ. Females are Z and W. When fish are raised at 22 degrees Celsius, ZZ fish develop into males, and ZW are females. However, if fish are raised at 28 degrees Celsius, the Z chromosome gets modified, so it's Z asterisk. So I'm going to say Z mod. So if you have Z mod W individuals, they are now males and that are fertile, and that they can pass on that Z mod chromosome to their offspring when their offspring are raised at 22 degrees Fahrenheit. So a cross between ZW and a Z mod Z male is in the Punnett square below. And the first question is, predict the pattern of phenotypic males. So how many males are going to look like males among the F1 offspring, and that's just their babies. So this guy's a male, this guy's a male, and this guy's a male. So that's a 3-to-1 ratio of phenotype males. Next question. At least one Z or one Z mod chrome is necessary for the survival of fish, or it's going to die. You can't be WW fish and die. It's very important that you get that fact before we move on to the next part of this question. Then it says, a researcher crossed 2 fish and observed a 2-to-1 ratio of males. How do you even get a 2-to-1 ratio? Like, you can't have one and have 4 fish present, because you've got 4 fish, so you could have a 3-to-1, you could have a 2-to-2, you could have a 1 to 1, but how do you get a 2 to 1 ratio? So we got to think through this. What is the male of the cross? So let's talk about our male possibilities. You have ZZ in a Punnett square, right? You have Z mod Z, or you have Z mod W. Now, which case are you only going to end up with 3 fish? So Z mod Z with a typical ZW female, you're going to have ZZ and ZW as your choices. This is going to be a 1 to 1 ratio. Z mod Z with ZW female, you're going to have Z mod Z, again, male, Z mod W, male, and Z, normal Z, male, and then normal Z, W, female. So again, this is our 3 to 1 ratio. That's what we just did there. Next, you've got Z mod W. So you have ZW is your normal female. This is our like crazy, we've made a male out of a female, or male. And so you have Z mod Z, male, Z mod W, male, ZW, female, and then WW. It told you that you have to have a Z to be a survivor, so now you have a WW fish, not going to live. So this is now a 2 to 1 ratio. So your male had to be Z mod W. Finally, it says, describe one fitness cost to the female mating with this particular male. So if you're a female and you mate with a Z mod W, 25% of your eggs, and fish usually lay a lot of eggs, you got to think about that. 25% of them now are not going to survive. They are not going to live. And that's a problem for fish. So while this does allow you to have more males in your population, so a wider variety of genetics passed on from one individual to the next, you do have an issue because after a while you're going to end up with 25% of your fish population dead. So I hope that's helpful.